I'm Kevin Kesseler, and I'm giving a talk on data analytics use case in pharmaceutical development. And this is something I've been working for the last almost 9 years to commercialize my postdoctoral research. And this is what I've come up with, and now I'm going to try and raise the money to actually get the software written and get clients and prove that all this works. So this is a use case, and it's apparently clipped off on the bottom of the screen, but there's a line at the bottom that is the most important line on this slide, which is, we do the math so you don't have to. So for any of you who were in the session on Wednesday of 8 hours of graduate level mathematics, don't worry about that here. Don't worry about there being a quiz. We're just I'm going to tell you how to cure cancer, and then you can go and do it for yourself on the way home. But easy talk, and we should be able to. I'll get out of here a little early. If you'd like to ask questions, I'm happy to answer them. Just yell them out. But that'll take more time then. I might not get out as early. Okay, so we have a use case, and hold on a second. Okay, that's better. In this case, I like to think about, because this is personalized medicine. This is about treating people based on who they are individually, making sure they get the right treatment for them. And a lot of people think DNA. You're doing personalized medicine if you're doing DNA. There's nothing more personal than that. But it really starts with and ends with the patient. And if you're not doing that, it's not really personalized medicine. And the loneliest person in the world is someone whose doctor has just said they had cancer. So they don't know anything. They just, their worst fear or one of their worst fears just happened. And they're looking at treatment and then this uncertain highway ahead of them that they may survive and they may not. So, there's the first thing is you have a lot of uncertainty, and that leads to fear. And what you need is knowledge, which leads to understanding. And knowledge and understanding, those are things as data scientists, we feel like we have a pretty good grasp at how do we wring knowledge and understanding out of whole buckets full of data. And I think any data project, any data analytic or whatever you want to call all this stuff we've been talking about the past couple days, is about a sort of intelligence operation. And so I'll get to that in a second. Didn't know the order of the slides. But let's talk about what we need to understand. So there's a lot of things that go into treating cancer. You have oncologists, dietitians, therapists, all sorts of things. But what can science bring to this? What do we need to get researchers involved in? How can they maybe bring cutting edge research to not necessarily curing cancer, but to helping cancer patients live longer? And one of the things we can do is researchers study biochemical pathways, how proteins interact that basically controls everything that goes on inside your body. And if we understand a pathway and understand that all these chemicals that pharmaceutical companies make, they make changes to one or more proteins that make a difference in how they function, and hopefully you find one that gets a desired therapeutic result, and it's it's helpful. And a couple examples of ways this might, understanding a pathway might lead to a pharmaceutical that gives a result would be DNA damage checkpoints. So DNA damage checkpoints are great. They protect you from cells that have damaged DNA from replicating, but chemotherapy causes massive damage to cells' DNA, hoping that they replicate and destroy themselves. And so the damage checkpoint, it stops the chemo from working. And so if you know how to model or understand how the checkpoint works, you can give a drug that knocks it out. And then the chemo just kills all the cancer cells, the normal cells aren't dividing, they have time to repair and survive. And so that's a drug that would help just any chemotherapy or radiation. Another would be, there's a class of proteins called connexins. And these are proteins that they do what it says on the tin. They connect cells to other cells. And one of the things that's true about all metastasized cancers is that their connexin pathways don't work. They don't have these cells. They don't get the message that they have neighbors, so they keep dividing. So and this is a huge pathway that is way beyond anything I've modeled before, but if you can understand this huge pathway enough to figure out what's going wrong in metastasis, well, you might be able to make a drug that turns it on again so that someone's metastasized cancer doesn't divide. Okay, so great. We'd like to be able to use research on these things to actually help patients, but how do we do the math? And again, no actual math in this talk, I promise. So, as I was saying before, I think any data project is an intelligence operation. So what you're trying to do is get actionable intelligence, and it's the same as if you're the CIA or Farmers Insurance or someone with a small company trying to do learning algorithms or something. So we have the data, and we want to turn data into information. We want to put it in a form that it's useful in some way. And then we want to get intelligence somehow. We want to get something that's actionable, that tells us something we didn't know before, or that somebody else is interested in. And so how do we do this? Well, first, we need to figure out how we're going to communicate, move these classes of stuff around. And so we have a kind of three things. We have metadata, intelligence, and information. So those are different kinds of data that's moving around. We also have feedback. We also have a system that's going back into itself and saying, this wasn't quite right, we need to make it better, or giving some information about the previous state. So that, I don't know why that's sideways. In the days when you have like a slide carousel, I can see how that could happen, but I'm at a loss. But anyway, if you look at it sideways, you see that we collect data. And we'll ignore the fact that's attached to intelligence for now, and ignore the fact that it's sideways. And thankfully, it's not sideways anymore. So we've got data, we can then mine it and get information and analyze it and get intelligence. But it's not just, we can't just collect any data and mine and analyze it. Um, well, first, collecting data it's slow. The data is always in horrible shape. You've got to vet it and clean it and whatever. But if we're doing computational modeling, we can generate all the data we want. We've got all the states and we can get whatever we want and be able to use a lot of clean data moving through our system. But then we also want to make sure that data is quality data, that whatever, generated or collected, we've got something that's not garbage in, garbage out. And then when we mine data, you can make a lot of statistics that are information, sure, but they're not information that anybody cares about, or they're not information that you can feed into some bigger analysis. So we want to make sure we get useful information, and then we want to analyze it for valuable intelligence. And valuable has a very specific meaning, which is it's worth something to somebody. Someone will pay for this. If someone's not willing to pay for it, well, it might be a fun project, but it's not a business plan. So now we want to set up an operation that does this cycle, and now we see that now the intelligence actually leads us to how do we generate more data, and gets us back to the start, and we can just keep going around this circle and generating useful intelligence. And of course, there's a road going off that you generate something and then you use it or sell it and make your profit. Now, we sort of what we want to put this around is a learning engine. The theme of the conference is symbiosis and automation. And this is sort of about what I'm trying to do here is first use this modeling to allow us to help pharmaceutical companies, but also use computational techniques to make the modeling more powerful and more effective. And so we have the symbiotic parts of this, and it's all built around a learning engine and to automate the process of scientific discovery. So we want to make the whole computer be able to actually find new drugs, find new, test new hypotheses for the researchers. Okay, so how do we do this? So after doing a model of a biochemical pathway that took about 5 years to go from conception to publication, I realized that's just too much. I was a PhD mathematician, and I had a lot of support. And in business, if it's not going to be done for 5 years, then we'll come back. Never. It's got to be done on time, and it's got to be done at a price that allows them to make money. So I made this software that it does all the math for modeling and helps you manage the complexity. And I could give additional hours of talks on that. But it's built around a model, an experiment, and a report. So it's essentially, we can think of it as a language or an object model that allows us to represent these three things and do appropriate simulations and move the information around. So, and these are all linked in. You're going to be seeing over and over again a series of arrows going around in a circle. And in the end, you get everything going around in a circle, and it hopefully, actually produces new new drugs to test that will help cure cancer. Okay, so what are the other agents that we're putting around here? What are the people involved, the experts? Well, we need theoretical biologists. We have a model. We need to know this protein interacts with this protein in this way, and we need to know those kind of things. And then we need the guys who actually, after we make a prediction or something, to go to the lab and test it and say, no, you got it totally wrong. We need those guys. So you need experimental biologists. But then you also have people in the pharmaceutical companies. They're doing research. They're trying to figure out, well, where can we, how can we put together all these chemicals we're coming up with to make new drugs that will make us money. And so the theoretical biologists are going to give the proteins and interactions that help us set up the model. The experimental biologists are going to, give us this. What is the experiment? You can't do a simulated experiment unless you're simulating an actual experiment that they can really test in the lab. Otherwise, you can't compare them. So how do you, what are the experiments you're going to want to do? What are the experiments you want to validate your model? And then the pharmaceutical companies, of course, have these big pharmacopoeias of different chemicals. There's all the chemicals in the public domain. And of course, they have what I think is one of the biggest assets of the pharmaceutical companies is they have all the data from the clinical trials and all this information about how these drugs work that even after their drug is out of patent, that's still valuable information. And again, valuable in the way the pharmaceutical companies want, it will make them money. So we want to, like I said, put this all around the learning engine. And what's that going to do? So with the learning engine, we can put all this stuff together and have these models virtually test hypotheses. So you've got some hypothesis about how these proteins interact and how that is, what are the important things that are making up the behavior of this biochemical pathway. And we can test it virtually and say, well, if your experiment comes out this way, it's this thing, and your hypothesis is probably correct. If the experiment comes out this way, it's probably the null hypothesis. So that helps the theoreticians who are, these networks get so big, their intuition fails. It's too big. You can't see how these are going to interact and what changing something's going to do, but you can do the calculations on the computer and see what happens. So then the experimentalists, you can give them dispositive experiments. You want to prove this? Okay, well, you do this experiment, and if the things are this way, this is what's going to happen. If the things are this way, this is what's going to happen. If something else happens, you've missed something in the model. There's something you don't understand. And so that helps them say, what are we going to do at the bench? Where are we going to focus our efforts? Just like later on with the pharmaceutical companies, they get promising potential combinations of drugs and later on we'll see how that gets sort of pumped out by this whole process. And then I made one change here. I said experiment, and now I've changed that to simulation. So model, simulation, and report. And you'll see why I want to reserve experiment in just a second, because, well, what we ultimately want to do, as I said, we want to automate scientific discovery. Well, how does scientific discovery work? You make a hypothesis, you test it with an experiment, you analyze it and update your hypothesis. So that's the cycle. If we can do, without having a human have to do, some part of it, then we've really automated scientific discovery. So first we want to validate these models. So I made a model of the G2 checkpoint or whatever system we're looking at. Well, why should anyone, let alone someone who's got cancer and is hoping you're coming up with something that's going to help them survive, why do they care? Well, you've got to validate it. You've got to make sure that, yes, this actually does match the data we have so far. So we've got some hypothetical model. We simulate it and simulate the experimental evidence we have, and then look at those results, look at the report, and say, yeah, this looks pretty good, or no, something's wrong here, and here's something else we can test, and again, go through the cycle until we get results that are validated. You set up the simulations, collect the information, modify the hypothesis. Okay, so we do this, and we end up with a model that is essentially a tool in research, right? These theoreticians, they can use this, and we have it, and we're worried about doing 10 million runs for a drug study, for a drug company, and doing 50,000 runs for a lab that wants to see, some pathway they're interested in. Well, we're great. We get to get the models that they're building. That gives us more valuable stuff, and we don't care about the computation time or the use of our software. So this gets both benefits on both the commercial side and the research side. So then we want those drugs. If this is going to work, it's not going to be cheap, and the only way to pay for it is if pharmaceutical companies spend about $5 billion for every FDA-approved drug. If you can't cut that price down, they're not even going to give you a second thought. So how do we do that? First of all, we've got our validated model, and we simulate it, and then we get some, and now we're simulating drugs. We've got the model working, so we know what the baseline is, what the control is, and now we can test combinations of drugs and see, does any of these have whatever effect we're looking for? And then we have the predicted effects that then go, okay, well, they inform our model and help us see, okay, well, how do we design a better drug? So we're gonna set up the simulations. That looks like the same slide as before. Anyway, so we do this, and we have we're testing, going through and testing the drug combination. So we want to tell a pharmaceutical company, instead of testing, these combinations of 3 or more of 1,000 drugs systematically, which they can't do, we just ran them all in the computer, and why don't you test these 10 at the bench? And so how do we get that and turn that into, like I said, automating the discovery process and really revolutionizing how we can develop drugs? So, like I said, we've got our experiment hypothesis, hypothesis experiment analysis. and they're our loop, and we go around that loop, and if we do it right, we get scientific discovery. This has been known for some time. And we just saw, we built our validation loop with this model and simulation and report, and we know that we can get a result that's defendable, it's validated, and we can run these simulations that help us in parts of our discovery. So what we want to do is put these two loops together. How we do that is with feed-forwards. Our experiments go to simulations. We need to simulate the experiments and figure out what the experiments to simulate are, and determine how we can't just have the simulations reflect back on the experiments without some feedback, or that the experiment came out differently. We've got to change our simulation. And similarly, we have an analysis that's got to go into a report. It's got to be that's our intelligence. It's got to be put in some form that the ultimate consumer of it can understand. The hypothesis updates our model. And then, of course, we have the other loop of it. And so we have everything's, all the symmetry. And that gives us our model and our validation loop, our infrastructure here, actually supporting and advancing our discovery loop. And the important thing about all of these is what stage of this does a human have to do? And the answer is the human has to set this up. The human has to set up the algorithms and like I said, all the infrastructure. But then, you push it, you let it go, you see if it finds anything. Maybe you're going to correct it and change change how the learning algorithms are working. But the actual scientific discovery process is fully automated. And I could go into detail about what each of these arrows is and what coding and technology would have to be done to implement it, or what stakeholders would have to be involved, and experts. But it's a known. Those are all known questions. Those are all things that people have done before, or the technology exists to do them. So I like to, like I said, focus on the patient and get back to patient zero, because that's all that matters is patient zero, the person in front of you who, what do they care about whatever I've been blathering on about for the past half hour. They have cancer. They want to know how you're going to help them with their cancer, and they want a team around them of the best people to help them fight their cancer. And so they live long enough to die of something other than cancer. And that's why I don't say I know how to cure cancer. Cancer is hundreds of diseases. Curing its way above my pay grade. But if someone with cancer lives long enough to get hit by a bus, does it matter to them that their cancer wasn't cured? No, it didn't affect their lifespan, as long as it didn't affect their quality of life. So just the quality, if you can improve the quality and duration of someone's life so that it's the same as it was before they had cancer, then I think, someone else can cure cancer later, but we're good for now. And so then Bob's your uncle, and you're done. So everyone now should know how to cure cancer, and go out and do it. Beat me to it. Then I don't have to. So thank you for your time, and if there are any questions, I'd be happy to answer them. Yeah?
Could you dive just briefly in some of the, the nitty-gritty of it all? Like when it comes to, you're talking a lot about like simulating pathways. Are you talking about like you have this protein pathway map and you want to say drug X gets knocked out? Is that the idea?
So you can do, and you can get good results just by analyzing the pathway. This protein is connected to this protein, and what I'm doing is a little bit more systems level, ground up. Is okay. These are the proteins. So I did, like I said, a G2 checkpoint model in my postdoc. There were 20 proteins in the model. Some of them were phosphatases, kinases, transporters, and it's just mass action kinetics and all the as good a representation of what goes on in the cell as we can make, because you need to have. You can't say this is the behavior I want. I'm going to program it in. If you don't program the interactions and then see if that behavior emerges, then you can't get this whole thing of actually getting scientific discovery. It's gotta come out of there. You can't bake your scientific discovery you want in.
So you start out with this accurate of a model of as much of the pathways as are relevant.
Or as much of the pathways as you think are relevant, and then when you're validating, you say, oh, okay, this matches my data, I've probably got everything important, or man, there's something, this just doesn't behave right. I just can't get this tuned, and there's got to be some kind of protein that's, I don't know. But yeah, you've got to do that, and you've got to make sure to bootstrap it up and have a solid model before you start using it to predict anything. Else? Hey.